I'm back with the last of the Module 5 lectures. So let's get into it. So this part of the lecture is talking about energetics, and in particular how cells obtain energy, and kind of the rules for energetics and the molecular machines, the biomolecular machines that actually do the chemical reactions, also called enzymes. So what we see here is a hummingbird feeding on some nectar. It is a collection of cells that all need energy to survive. So that hummingbird is at a very high metabolic rate. And so we're going to start to explore how energy behaves in living organisms. All right, so the first part of this, we're going to try and answer the question, what are the rules of metabolism? And then the next part is to ask ourselves, how do living organisms accelerate biochemical reactions? The short answer there is enzymes. And we'll talk about the catalytic cycle of enzymes, the effects of environmental conditions on enzyme activity, and then regulation of enzyme activity. All right, before we can even start talking about metabolism, or really the rules, we have to think about what are we talking about? Essentially, we're talking about transformations of energy. And there's a lot of different types of energy that we could think of, like chemical energy or gravitational energy or electrical energy or nuclear energy. But they all fall into two big categories. And those big categories are either potential energy or kinetic energy. So potential energy is the potential energy is stored energy. It is energy that is available to do work at some later time. So if you think about this waterfall here on the right, the water at the top has potential energy because of where it is in relation to the center of the Earth, or the center of mass of the Earth, if we want to get really technical. And by being a little bit higher up than the surrounding water, that is gravitational potential energy. And then as that water falls, it is converted into kinetic energy, sound and motion and heat at the bottom of the pool here. In a similar way, this ice cream cone might represent potential energy, energy that is stored. And when a young human eats it, they might be able to use that energy to ride their bikes around or walk around or think. So those are just kind of the two basic types of energy. All right. Oh, no, we need to get rid of our things. Okay. So there's some rules that govern energy transformations, and these are called the laws of thermodynamics. So we aren't going to do a really deep dive of thermodynamics. If you're really into that sort of thing, go take a physics course. But basically, there's two laws here. The first law is conservation of energy, which states that energy cannot be created or destroyed. It can only be transformed from one form to another. So in less technical terms, you can't win. There's no free lunch. There's no free energy. There probably isn't nuclear fusion at room temperature. If anyone tells you otherwise, they are probably selling you something, and you probably should not trust them. The second law of thermodynamics concerns efficiency. And there's maybe a couple different ways to state this, but essentially the technical term that we are going to use, we're going to say that the entropy in a closed system is always increasing. So if no energy can get in or out and no molecules can get in or out, the disorder, the random motion, the chaos of a closed system is always going to increase. And another way to think about this is that energy, as it gets transformed from one form to another, some of it is always going to be lost as heat, as this increased entropy that you can never recover. So another way to put this is that you can't break even. Efficiency is never 100%. It's always going to be less than 100%. So there's maybe a little loophole in this statement. If you could get to absolute zero, you could possibly build like a perfectly efficient heat engine. Because the problem with heat is you always have to find some place to dump it when you use that as your source of energy. And if you had absolute zero as a place to dump your heat, woohoo, you could break even, except that nothing is ever absolute zero, because where is it going to dump its heat to something even colder? No, there is no colder. So anyhow, those are the rules that we have to play by. And this is true for the abiotic parts of an ecosystem and also the living organisms in an ecosystem. So what does this mean for living organisms? Well, organisms have to capture energy from somewhere else in order to live. It's just a fact. And maybe it's an inorganic source like the sun, or maybe it's an organic source like some other unfortunate organism. The ultimate energy source for all life on Earth, well, it can't come from within the Earth, really. That's a closed, if we considered Earth as a closed system, we wouldn't have billions of years of life and evolution. There's just not enough energy to make that happen. So instead, that energy comes from somewhere else, namely the giant nuclear reactor in the sky called the sun, which provides basically, for our purposes, it's kind of free energy. But eventually the sun is going to spend all its nuclear fuel and burn out, and life can no longer exist on Earth sometime in the future. So, all right. So the sun is passing its energy along to the producers, the plants of an ecosystem, and a lot of that energy is actually being lost as heat and not being captured into the biosphere, or I should say the biological community. And then some of those plants are consumed by animals, the consumers, and what isn't consumed by animals or other consumers ends up in the decomposers, and eventually the decomposers will get any leftover energy from the consumers as well as the plants. So, and then ultimately, this all ends up as heat. Now, because of this, can food chains be infinitely long? The short answer is no. There's an inefficient transfer of energy at every step. So at some point, it's going to be so efficient that it just doesn't pencil out for a larger predator to be consuming some other predator. So, and we'll visit this again when we talk about ecosystems. All right, so why do we need to know about these rules of metabolism, rules of energy transfers? Well, really, we want to talk about chemical reactions. And in a chemical reaction, the reactants combine to form one or more products. And a good example of this, or maybe a terrible example of this, we saw in the explosion of the Hindenburg where hydrogen and oxygen combined to produce water and a giant fireball. So this is an example of a chemical reaction. And it's a particular type of chemical reaction where we get energy out of the reaction. So this is actually maybe a useful one for us in the future when we're thinking about how to power our societies. Hydrogen is combining with oxygen releases a lot of heat, and it's pretty efficient. So, and we can split apart water if we have a renewable energy source into hydrogen and oxygen. And yeah, that's not going to be 100% efficient, but it is actually ends up basically being kind of like a really good battery. But there's a lot of technological hurdles, and we don't want the Hindenburg to happen again. So anyhow, all right, so these two main types of chemical reactions are either endergonic, meaning energy comes in, and so the products have a higher energy level than the reactants did, or it could be an exergonic reaction, where the reactants had a higher energy level than the products did. And in the process, energy was released. Okay, thinking back to the burning of hydrogen, is that endergonic or exergonic? Yeah, that's going to be exergonic. So that is one where energy comes out of the reaction. Okay, so another thing to think about is activation energy. So these chemical reactions have to get started somehow. And so there has to be some small input of energy. And if we think about burning a match, I'm actually going to do this here because I'm in the labs and I've got some water to put it in. I'm going to briefly stop the sharing. So you can see, so this is a matchstick. It's made of wood, which is essentially cellulose, which is essentially sugar, right? There's a little bit of phosphorus on the tip of the match. And if we let this match sit here for a very long time, nothing's going to happen really. Maybe if we got it wet and some termites found it, they would eat it, but on its own, it's not going to do anything. So we have to supply some activation energy for the wood to start oxidizing in the presence of oxygen. So basically we're going to combust wood, we're going to burn glucose or cellulose, which was made of glucose monomers. We're going to burn that in the presence of oxygen, and with a little bit of heat, a little bit of activation energy, we can get this reaction going. Maybe. Okay, maybe we can't. Try a different match. These have been sitting around for quite a while, so we'll see. There we go. So this is a chemical reaction. We know it's reacting because there's an energy transformation. There's an energy release. The stored energy in those sugars are turning into carbon dioxide and water and releasing heat, maybe a little bit of sound. So that's an example of a chemical reaction. I'll go back to sharing this screen. All right, so really if our reactants is the cellulose or the glucose essentially in that matchstick, and the products are carbon dioxide and water, there's some activation energy that we need to supply in order to get this reaction going. And then once it goes, if it's exergonic, the energy that it releases in that reaction supplies activation energy to burn the next few sugar molecules. So yeah. Okay. What about catalysts? How do they help chemical reactions? Catalysts are important because they speed up chemical reactions by lowering the activation energy. So that match has to get really, really warm for it to start to burn, right? But if we had the right sort of catalyst, we could maybe get that reaction to occur at a much lower temperature, much less extreme conditions. So catalysts can speed up endergonic and exergonic reactions. Although with endergonic reactions, we still need to supply the energy somehow. So an endergonic reaction is just not going to run unless there's a supply of energy. That energy difference has to be accounted for. Catalysts are also not consumed or permanently changed in the process. So at the end of the reaction, they are the same as they were at the beginning. And enzymes are essentially just biological catalysts. So they help a chemical reaction to occur without being used up. So here, if we look at our reaction graph, without a catalyst, there's a lot of activation energy needed. And if we have a catalyst such as an enzyme, the activation energy is much lower. Okay, so let's learn about enzymes. So essentially, all enzymes are proteins. Okay, am I clicking on that? Yeah. There's a few exceptions to this, and actually they're ribozymes, which are made of RNA, but I almost wish I hadn't just said that, because for our purposes, we can think all enzymes are proteins, and we will be right 99.99% of the time. Okay, enzymes can only catalyze exergonic reactions. And I should say the net reaction that an enzyme catalyzes has to be exergonic. There might be some endergonic steps where enzymes can couple an exergonic with an endergonic reaction to make something happen. But for the most part, the net reaction has to be exergonic. Enzymes can participate in chemical reactions, but aren't used up in them. So this is kind of the definition of a catalyst. And enzymes greatly increase the reaction rates. So eventually, given like probably hundreds of years, oxygen will start to oxidize the cellulose in this matchstick. But we could burn it with very high heat very quickly. Or if we want it to occur similarly quickly, if we applied the right enzymes, we could maybe make this happen. Okay, so essentially, if we have a chemical reaction where the reactants become products, the enzyme, we usually write the name of the enzyme over the reaction arrow to indicate that it's participating but not being used in the reaction. So it's neither a reactant nor a product. It's a catalyst, right? When we're talking about enzyme catalyzed reactions, the reactants have a special name called substrates. Sometimes there's only a single substrate, sometimes there's multiple substrates that join the enzyme and allow the formation of the product or products. Okay, so how do enzymes work? Essentially, there is a cycle of catalysis or a catalytic cycle. So here is our enzyme. And there's a very special spot on the enzyme called the active site, which is where the substrate or substrates bind. And the active site is shaped so that it will fit the substrate. And it also has just the right chemical groups around the substrate to hold it in place and to help the reaction occur. And again, these enzymes are big old proteins, so it's got that 3 or 4 levels of protein structure So it's folded up in a very special way to get particular amino acids right next to the substrates. Okay, when the substrate binds, that forms the enzyme substrate complex, and the enzyme will change shape very slightly as that substrate binds. Because of this, that might put a little stress on particular chemical bonds, or it might encourage the formation of different chemical bonds. But the net result is that these substrates get converted into products. And now we have an enzyme product complex. And the products don't usually fit quite as well into that active site. So they are released. So the products go off and do whatever it is they're going to do. And the enzyme returns to its original shape. So it can actually go back and pick up a new substrate and repeat this cycle over and over and over again. A single enzyme can probably catalyze tens of thousands, if not hundreds of thousands of reactions without degrading. All right, so here's maybe a fancier picture of that idea. So I just like this because the enzyme is more of a blob of a protein. And we have the active site with two different substrates that are going to bind. As they bind, the shape changes a little bit. And that encourages the reaction to take place. And then some products are released. All right, and then the enzyme can go and bind the substrates again. Okay, so that's how enzymes work. How are enzymes regulated? What things can affect the rate of enzyme catalysis? Well, if we remember back to our discussion of proteins, we talked about the different things that can denature a protein, and anything that can denature a protein can affect the activity of an enzyme. So these could be environmental factors like temperature or pH or salt or solvent polarity. So any of these things could cause an enzyme to work better or worse. And usually if it's changed too much from the ideal, the enzyme will not work at all. As an example of that, enzymes are very sensitive to temperature. So as the temperature increases, molecules move around more, right? So that means that the substrate and the enzyme are going to meet up more often, which is good, right? Except that as the temperature increases, the denaturation of that enzyme, which is a protein, which is one type of protein, I should say, that denaturation is going to increase. So the percentage of enzyme that is still functional starts to decrease, and it happens pretty drastically. It's a pretty abrupt cutoff from, oh, the proteins are folded just fine to, oh yeah, those proteins are totally denatured. And so usually there's an optimal temperature for any given enzyme. A lot of human enzymes have an optimal activity somewhere around 37 degrees Celsius, because that is our normal operating body temperature. If we look at hot springs bacteria, their optimal temperature might be 80 or 90 degrees Celsius. So it just kind of depends on the organism and its environment. And there's other organisms that I can think of that they will die. They mostly live around on snow and ice and glaciers and things like that, and you pick them up and the heat of your hand will kill them because you've denatured all their enzymes. So there are a wide variety of adaptations for different temperature environments. So that's one example. A different example is pH. So if we look at the enzymatic activity for different pH values, we find some different patterns. So for typical human enzymes, our pH is somewhere around 7, maybe a little bit basic, actually. Maybe 7.35 is the number that comes to my mind, but I could have that wrong for like blood pH. But anyway, it doesn't matter. The exact value is somewhere near 7. And so a typical human enzyme like the salivary amylase that is working in our saliva to digest starch into simpler sugars, that is going to have an optimum activity at pH 7. And if you go to like pH 5, there's really not much activity, and you go even more acidic, there's no activity. And then as things become more basic, same sort of pattern, right? Okay, well, what about the pepsin, which is a different digestive enzyme that lives in your stomach, which is a very acidic environment? The optimum pH for pepsin is maybe around pH 2. So that's the usual pH level of your stomach. And that's going to digest proteins at that pH. And then what about trypsin? In your small intestine, the pH kind of swings in the other direction, and it's a little bit alkaline compared to your blood or the rest of your body. And so the optimal pH for trypsin might actually be like pH 8 or 9. This is showing maybe pH 10 or 11. There's probably an enzyme out there that has that optimum, but this is just kind of simplified for clarity rather than the actual values. Okay, so enzymes are very important for all the different chemical reactions you need to do, like digesting your food or building your own proteins, or basically anything inside the cell is happening because of the result of an enzyme sooner or later, right? A lot of times we can't have a single enzyme that can accomplish a radical change from a substrate to the final end product, right? So instead of doing this in one big step, we have to do this using multiple enzymes in a lot of little steps. So the substrate goes to enzyme 1, and that transforms it into the first intermediate substrate, which then can go to enzyme 2 and then get transformed into the second intermediate substrate. That goes to enzyme 3, which actually makes the final product. So this happens all the time. There's a lot of steps in cellular respiration that we will talk about in a couple of weeks, or photosynthesis. These really complicated biochemical pathways get broken down into simpler steps. The really great thing is that if we embed these enzymes into a membrane, we've kind of linked that path together, and the substrates and the intermediates don't have to travel very far to find the next enzyme, which can really increase the efficiency. And so this is why eukaryotes have so many membranes. For doing complicated biochemistry, we can encapsulate it or embed it into a membrane, and that will really speed up the rates of reaction and allow us to do really complicated biochemistry. Okay, so this is a biochemical pathway. And a lot of times in these biochemical pathways, we have some inhibition. And really, what this means is that the concentration of end product needs to be kept at some optimal level, right? So we don't want none of it, and we don't want too much of it. We've got to regulate somehow. And one way that this works is called feedback inhibition. And in feedback inhibition, the end product is actually binds to the first enzyme in the whole process, and it binds in such a way that the active site gets changed so that the substrate doesn't bind. And that will kind of shut off this whole biochemical pathway until the end product starts to get used up, and then there's not as much around, less of it to bind with those enzymes, and then they can start binding substrate again. So this is kind of a clever way for cells to regulate the activity of enzymes. There are other ways a cell might regulate an enzyme's activity. One very common way is just to simply switch the genes that code for these different enzymes on or off. So by allowing the gene to be read and built into proteins that will activate that enzyme, and after a while that protein will break down, and if the switch is off for those genes, then the enzyme isn't going to be produced. Another way, which is pretty common, is that a lot of times the enzymes are first synthesized in a form that isn't active. So they have to be kind of activated somehow before they start working. So they're present. We don't have to wait for protein synthesis to occur, but we do have to wait for the right signal to activate those enzymes. A lot of times there can be inhibition from other molecules, kind of like the feedback inhibition that we just showed you, which would be, we call it non-competitive inhibition, because the inhibitor doesn't bind to the active site, but it binds somewhere else on that enzyme and doesn't allow the active site to fit the substrate any longer. Competitive inhibition, usually we're talking about drugs or poisons that actually compete with the substrate for access to the active site on that enzyme. And when that happens, there's kind of depends on the balance of substrate and inhibitor. If inhibitor is more concentrated than substrate, that's going to be the one that binds, and vice versa, right? So the dose is very important when we have this competitive inhibition. And lastly, there's allosteric regulation, which usually we see that with non-competitive inhibition. And allosteric just means other shape. So, and usually there's an allosteric site that is away from the active site, that when something binds, we can have the active site in a slightly different shape. And this allosteric regulation is kind of interesting because we can have allosteric inhibition, where the enzyme is normally active unless the inhibitor binds, and then the active site isn't the right shape anymore. Or we could have allosteric activation. So normally the enzyme is non-functional until the activator binds. Some other molecule binds to the allosteric site, and that activates the enzyme and causes the active site to be in the correct shape for the substrate. So both possibilities could happen for any given enzyme. It's usually one or the other, not both at the same time. So kind of wish they had different enzymes in this drawing. Whew. So that was a lot to get through. We'll talk about this more, and I think that is it. So until later.